Hi, my name is Ariel Munitz, and I will present some of our data that was published in the manuscript entitled Mouse-Resident Lung Eosinophils Are Dependent on IL-5. Eosinophils were traditionally considered terminally differentiated cells with little to no heterogeneity. They mature in the bone marrow, migrate into the blood, and then transfer into various tissues where they can acquire unique phenotypes in response to diverse stimuli. Recently, two subsets of eosinophils, namely resident and inflammatory eosinophils, were described in the lungs of allergen-challenged mice. They were broadly termed R-Eos, representing resident eosinophils, and I-Eos, representing inflammatory eosinophils. These two cell types can be potentially differentiated by their relative expression of surface markers such as Siglec F, where R-Eos are Siglec F low and I-Eos are Siglec F high. In addition, previous reports suggested that R-Eos are independent of IL-5, whereas I-Eos are IL-5 dependent. This distinction is of particular interest since it may have major therapeutic implications. Neutralization of IL-5 through anti-IL-5 cytokine therapy may deplete the I-Eos and keep R-Eos intact. This is potentially good since R-Eos were attributed with anti-inflammatory immunoregulatory activities. Conversely, depletion of eosinophils using antibody-dependent cell cytotoxicity mechanisms through targeting anti-IL-5 receptor alpha may deplete both populations, and this is bad because this may deplete a population which suppresses inflammation. In this study, we hypothesized that resident eosinophils are actually dependent on IL-5 for their functions. In order to assess this, wild-type mice were challenged with house dust mite, and the presence of lung eosinophils was determined. At baseline, one population of lung resident eosinophils were identified, which were Siglec F intermediate and CD101 negative. We could not identify in any of the lung eosinophil populations the expression of CD62L, a suggested marker of resident lung eosinophils. On day 10, two eosinophil populations, which appeared to correspond with the previously identified R-Eos and I-Eos, were identified. However, by day 21, most lung eosinophils consisted only of Siglec F high cells and expressed CD101. This actually suggested that Siglec F expression is upregulated on eosinophils and that an environmental factor such as IL-5 can regulate Siglec F expression. To assess this hypothesis, whether IL-5 regulates Siglec F expression, eosinophils were obtained from the peritoneal cavity of wild-type mice and incubated with IL-5. IL-5 increased the expression of Siglec F in a concentration-dependent fashion. Similarly, eosinophils from the bone marrow, blood, spleen, and lungs of IL-5 transgenic mice displayed elevated levels of Siglec F expression. Administration of anti-IL-5 neutralizing antibodies to naive and day 10 allergen-challenged mice decreased the expression of Siglec F in the peripheral blood and lung eosinophils from naive mice, as well as in eosinophils from the blood, lungs, and bronchoalveolar lavage fluid of allergen-challenged mice. Conversely, administration of IL-5 to the peritoneal cavity of wild-type mice increased the expression of Siglec F in these peritoneal eosinophils. Collectively, these data suggest that Siglec F high eosinophils in the lungs of allergen-challenged mice may comprise a mixture of both resident and inflammatory eosinophils, where resident eosinophils merely upregulate the expression of Siglec F. Furthermore, neutralization of IL-5 in naive mice markedly decreased the percentage and total numbers of resident lung and peripheral blood eosinophils. Furthermore, IL-5 neutralization decreased the levels of blood, lung, and BALF eosinophils in allergen-challenged mice. These data show that anti-IL-5 treatment downregulates Siglec F expression and depletes all population of lung eosinophils. These data are inconsistent with the finding that resident eosinophils are independent of IL-5, and they further suggest that distinct eosinophil populations in the asthmatic lung represent a continuum of activation states rather than different cellular subsets. I wish to finish by thanking my entire laboratory, and especially Avishai Dolitsky, who conducted the work, and to all of our funding agencies who made this work possible. Thank you.